For our entire history, we've built things from wood, stone, and metal. But what if we could actually learn to speak the language of biology? What if we could start building with the very same stuff that makes up life itself? Well, today, we're going to dive into exactly that.
And no, this isn't some far-off sci-fi concept. This is the big question behind one of the most game-changing fields out there today, protein engineering. It's all about taking nature's own microscopic machines and, well, redesigning them to do brand new things for us.
So here's our game plan. We're going to start with what these tiny machines even are, then figure out why we'd ever need to improve them. After that, we'll walk through 3 incredible ways scientists are doing just that. And finally, we'll take a peek at what the future of all this looks like.
All right, first things first, let's meet the hero of our story today.
Proteins. These things are the absolute workhorses of every single living cell. It's best to think of them as these tiny biological nanomachines. They are doing, well, pretty much everything, from giving your hair its structure to digesting your lunch to fighting off viruses. They do almost all of the heavy lifting in the biological world.
And that brings us to a really important question. If nature has spent billions of years getting these proteins just right, why would we need to change them?
Well, the answer is that the world inside a cell is, it's pretty cozy. It's carefully controlled. The world inside an industrial factory or a lab, not so much. See, natural proteins, or enzymes as they're often called, they just didn't evolve to handle crazy high heat or extreme acidity or all the other harsh conditions we find in manufacturing. We're talking about making medicines, biofuels, even the stuff in your laundry detergent. The bottom line is, we need to make them a whole lot tougher. So how in the world do we do that? Well, the first major strategy is to take a page right out of nature's playbook and then just hit the fast-forward button.
This is called directed evolution, and it's basically like breeding proteins. You start with a gene, and you intentionally create a bunch of random mutations. This gives you this massive library of slightly different protein versions. Then, the real work begins. You screen that library to find the one that does the job best. That's your winner. And that winner? It becomes the parent for the next round of mutations. You just keep repeating the cycle, getting a little better each time, until you have a protein that's perfect for the task.
But here's the catch with directed evolution. It's incredibly powerful, but it's a total brute force approach. You're basically searching for a lost key in a gigantic dark room. You're making millions of random changes and just hoping you stumble upon the right one. It's a pure numbers game.
So, that raises the obvious question, right? Can we be smarter about this? Can we stop fumbling around in the dark and maybe draw ourselves a map? Well, that is exactly what our next method is all about. This is where rational design comes in. Instead of just random guessing, this approach uses what we already know about a protein's 3D structure and how it works to make very specific targeted changes. We're not creating a massive library of random shots in the dark anymore. We're creating a small, super focused library based on an actual educated guess. And believe me, this isn't just theory. It has a massive real-world impact. A perfect example is how they manufacture sitagliptin, a drug for type 2 diabetes. To make it efficiently, scientists needed a really good enzyme. It took 27 specific changes to the amino acids, over 11 rounds of engineering to create the high-performance enzyme they needed for production on an industrial scale. Can you imagine trying to find that exact combination with a purely random approach? It would have taken forever. And this is where things get really cool, because this shows you the true power of modern protein engineering. The scientists didn't just pick one method. Nope, they combined them. They used rational design to intelligently tweak the most important part of the enzyme, and then they used directed evolution to fine-tune the rest of it to make it more stable. Best of both worlds.
Now, the tool that makes all of this intelligent design possible is computational protein design. It's basically a virtual lab inside a computer, where scientists can build models of proteins and predict how different tweaks will affect them, all before they ever have to pick up a test tube. It saves a huge amount of time and money.
So how does it actually work? Okay, think of a protein like it has a skeleton, or a backbone. The computer keeps that backbone locked in place. Then, the software starts swapping out the little bits that stick off of it, the side chains, and for every single combination, it calculates the energy. The computer follows some pretty simple rules. It likes stable bonds, and it hates it when atoms get too close and clash. The design with the lowest energy score is usually the one that's going to be the most stable in the real world.
Okay, so we've seen how to tweak proteins that already exist.
But what if we could take the ultimate leap? What if we could design and build a completely new protein, one that nature has never, ever seen before?
This is the final frontier, de novo design. This isn't about remodeling a house anymore. This is about being the architect and the builder for a brand new house, from the ground up, designed to do something totally novel.
This might sound like it's straight out of science fiction, but it's actually happening right now. So, for example, could scientists use this approach to create a totally new protein that's specifically designed to go after the influenza virus? And the answer is a huge, resounding yes. Researchers have already done it. They successfully used de novo design to build a brand new protein from scratch that targets and shuts down a key part of the flu virus. This is a massive breakthrough. It shows we can actually design custom antiviral weapons from the ground up.
So what does this all mean for the future? Well, we've clearly gone from just being observers of nature to becoming active participants. We're redesigning it.
You know, this quote from the researchers really says it all. The future isn't about picking one method over the other. It's about combining them, using computers to dream up new designs, and then using experiments in the lab to test and perfect them. This lets us create powerful new medicines and tools that nature, all on its own, would never have even thought of.
It's kind of amazing when you step back and think about it. We've gone from just being readers of the book of life to actually becoming writers. Nature gave us the alphabet, the amino acids, and we are finally learning how to arrange them into new words, new sentences, new stories. The only question left is, what should we build next?